Hi, this is Dr. J. Bailey, and in this video, we are going to go through topic 3.2, which is all about chromosomes. So, here are the main target things that we're going to talk in this video. We're going to talk about the prokaryotics and all about its chromosomes, it being circular. We're also going to talk about its plasmids, which eukaryotics don't have. Then we're going to talk about the eukaryotic chromosomes, how it's linear, and it has to do with histones. We're also going to talk about different species of eukaryotics with having different amount of chromosomes and different genes. We're also going to talk about the homologous chromosome, which is very important when it comes to meiosis and mitosis, if you want to say more meiosis, of course. We're also going to talk about the difference between diploid and haploids and how do they work, in which situations do you see them. We're also going to talk about how the number of chromosomes and what this has to do with different members of species. We're also going to talk about karyogram, which basically is showing the chromosomes, organize them according to their length. We're going to talk about sex chromosomes as well as autosomes. And we're going to talk about Cairns technique and how they measure the length of DNA with autoradiography. And we're going to also compare some genome size of different types of species. We're also going to compare the diploid chromosome numbers of different species. We're also going to learn how to use a karyogram to figure out if there's any kind of sex, if it's male or female, or if there's any kind of diagnosis such as Down syndrome. And last, we're going to look at some databases to identify some human gene as well as polypeptide products. So, with that, before we can even start, we have to have some definitions in our heads. The first one is about genome. So genome here basically means the entire genetic information of that one organism. So it's like you and all the genes information that you have inside of your body. The genome size, that is the total length of DNA inside of you. So it's be careful between the word genome size and genome, as genome size is more of the length. So that's the difference here. One is the length and the other one is genetic information. That's the difference between those two. When it comes to gene, that's a heritable factor that controls a specific characteristic, in the case could be trait. So gene is the one that is being passed on from the parents to the child. Now inside of the gene, you have the allele, which is a specific form of gene. And you can have so many different types of alleles. So some of them have more bases than the others, and we're going to see exactly their location, which is known as gene locus. So in here, we have basically the chromosomes inside the nucleus. If we put them out, you can see you have one full chromosome here. And in that chromosome, you can see that the sections of it, these little sections in dark here, that are being highlighted as green, are different genes. And inside each of the genes, you have a certain length of DNA inside of it, and that represents the gene. So, moving on, let's look at the difference between eukaryotics and prokaryotics. When it comes to the number of chromosomes, we know that prokaryotes have one, while eukaryotes has at least two or even more. Like humans, they have 46. When it comes to shape, the prokaryotics usually are circular, so they usually have the turn like a circle, while eukaryotics, they usually are more linear. When it comes to histone, in this case, the histones basically are proteins. And it's not found in prokaryotics. And the reason why, because the prokaryotes are just small like this. But when it comes to eukaryotics, they're so huge. So what they do is they take a histone, which is basically a little circle like this. And what they do with the DNA is they wrap it around that histone. And they can keep doing many histones like that and wrapping the DNA. So imagine you're having more and more histones here. And what do you do with the DNA? You just simply wrap the DNA around like this. This is where you see the shape of the chromosome, which is an X. So that can only be found in the eukaryotics. You do not need the prokaryotics because it's way more smaller. Now, when it comes to plasmids, it's mostly found in prokaryotics. And plasmids are basically DNA, but it's basically in the shape of a circular. When eukaryotics, you never see any plasmids in the eukaryotics. And when we talk about organizing into pairs, because it's only one, so there's no organizations of pair. However, eukaryotics, you're going to see most of them are pairs. So most of them is like 46, 24, and so forth. So it's one of the questions that many come about these two is knowing the differences. So this chart is extremely important that you remember this chart, because this is enough information that you can get points if it says distinguish between eukaryotics and prokaryotics. So moving along, Now we're going to talk about the species chromosomes. Now, when you look at this chart, you can see the interesting about it is how we have fruit flies has 8 chromosomes, the mollusks have 80, the toads have 22. Now, looking at this, I give you a few seconds just to look, and you can see right here that human is 46. Now, if human is 46, think of any species here that have a close number to 46. Now, if you notice, chimpanzees are the ones that are closest to the humans, right? They have 48, so that's very close. Now, that's why we think that there's a similarity because of them have closer numbers, but doesn't necessarily mean that they're same. Because if you look at the amoeba, which has nothing to do with humans, right? It's just basically a very straight like this picture of a amoeba, and it has nothing to do like us. It's more like a bacteria that is moving around. And you can see that these two are very close in numbers. So it does not have to do much with being, if they have close numbers of the beings, then we're going to be like each other. Because you can even see here that wheat is 42. It doesn't mean that they actually have the same thing, right? Now, you can look from all of these and you can see the crayfish right here has 100, more than 100. And crayfish is way smaller than humans. So also, the number of the chromosomes does not tell the size, the big size of the species whatsoever. So it's important that you know that, yes, the number of chromosome will tell you which species it is. But it doesn't mean that if you have more chromosomes, that means you're bigger or you're more complex. That's not true. So bigger numbers of chromosomes, it's only about telling which species you are. But big numbers of chromosomes does not make you more complex and does not make you bigger. So here is basically the main ideas that we can have. We said different species have different amounts of chromosomes. As you can see here, there's about 16 of them showing different numbers. And different species also have different amount of genes in base sequence. So this is one thing we know. This is for sure. Every species have their own number. Every species have their own genes in base sequence. However, this clearly shows you that it doesn't mean that if you have more chromosomes, that means you're more complex or you're bigger. It has nothing to do with that. Here we talk about the genome size. When we talk about genome size, we're talking about the base pairs. That means the A, T, C, and G. You can see that humans, for example, they have 3 billions. And they have 46 chromosomes and about 25,000 genes. Now, interesting about this, you can look at, for instance, a mouse. A mouse has 2.9 billions with 40 chromosomes and 25,000 genes. So it doesn't necessarily mean that if you have the same chromosome number that you're going to have the same amount of genes or the same amount of base pairs. You can look at the bacteria, for instance, here. It has 4.6 million, and it's just one chromosome. It has 3,200 genes. But look at the interesting part that it's the same amount of genes as human is the plants. The plants here, they have 25,000 genes, just like the humans. However, they only have 10 chromosomes, and they only have about 157 million of base pairs. So you can see that there is no such thing as a link with the amount of numbers of base pairs or the amounts of genes or anything like that, with complexity or anything. So what really matters is what's inside of those genes, the size of the estimated size of the base pair, and as well as the sequence. Most important is the sequence, because the sequence tells you what structure and function you are. So remember that the sequence is the most important thing. So we're looking at here. We have the T2 phage, we have the E. coli, we have the Drosophila, we have the Homo sapiens, and the Paris japonica. So basically, it's basically saying a virus, a bacteria, fruit fly, human, and kind of place. So one of the things I'd be asking you is for you to look and compare the genome size. And again, when I say genome size, we're talking about the length of the DNA. That means the A, T, C, and G, how many did they have? If you look at the virus, it has 170,000 BPs, and then it's very tiny that we can even see. But then you have a plant that is 150 billions BP, which is a very small plant. But look at compare the plant to the human. Human only has 3.2 billion. So just from here, look at the number. It's way more bigger in the plant than compared to the human. So basically saying that the genome size of a plant is bigger than the human. And this just proves again that it's not the amount that you have of bases that will say that you're more complex or you're bigger. It does not have to do anything with that. But we do know that the chromosome number will give you a certain species. So if there is a 46, obviously it will be a human. If anything is 48, it's chimpanzee. If anything is 78, it will be a dog. If it's any 24, it's a rice. So the number of the chromosome will tell you which species is. But the size of the chromosomes, the amount of genes, or the amount of base pairs has nothing to do with anything with the size or complexity of any species. So here we have this is photo came from BioNinja, and here it's showing different types of species here in the bottom. And here in blue we have the size of the genome, which means the A, T, C, and G, and the number of genes. That means genes, for example, in humans there's about 25,000, and the diploid numbers basically means the amount of chromosomes they have. Now, look at the interesting part here about the amount of genes. We can see that the rice here has a lot more genes than even the humans. But again, the ones that have the highest amount of diploids in this case will be the humans, as you can see. And then you can talk about the size of genomes, and you can see that the chimpanzees have a bigger size of ATCs, while the bacteria here has the smallest one. So this is just comparing three things: the amount of ATCGs, the amount of genes, and the amount of chromosomes. And from all of this, you can see is that it's completely spread around, and there is no logic, there is no correlation whatsoever with the numbers of chromosomes, genes, or genomes with anything to do with the bigger size or the complexity. When I say bigger size, I'm talking about tall and short. And complexity, I'm talking about having such a complex system like the organs and stuff. So the only thing that matters in all of this process is the amount of chromosomes that they have. So every species have a specific number, and it's always like this. When I say that, I'm saying humans will always be 46. And that means if you have a 46, that's a human. Of course, you might have a 47, 45, but then this human will have issues in their body. And we can also talk about the virus. If the virus only has a chromosome 1, it will always be chromosome 1 is a virus, and so forth. Now, where do we find these genes? When we say the word gene loci here, it means where is the position of that gene in that chromosome. So here you see a chromosome, and you can see that the gene is located right here. So this means this is the loci, this is where the gene is located. Now, inside of the gene, we have alleles. Alleles are basically the form of a gene, and they are the ones that tells us what kind of traits the gene will present. So in here, going more specifically, how can you separate or differentiate an allele that has a capital letter and an allele that has a small letter? It has to do with the nucleotide sequence. Here you can see that both have a very close sequence. However, there is a difference here when it comes to the letter. There's a switch going on. So what makes one allele different than another allele? It is definitely at the base sequence, right here. That's what makes the difference between the big A and the small a. It's one little switch in one of the nucleotides in the DNA sequence. You can also see it here. The differences is all about the nuclear base. So if there comes a multiple choice saying what makes one allele different than another allele, it's all about the nucleotides, or should I say one base. Now, since we're talking about chromosomes, and we know that there is a lot of replication happening in cells, that's when you make an extra copy of DNA. We also know that when you make an extra copy, here you have that one copy only of DNA, and then before you go through mitosis or meiosis, that one copy here will create a second copy for it, and that second copy will be attached by the centromere. So that means here, these two, we know it as cystochromatid. So this is copy of that copy. So this is usually what happens before you go towards mitosis. This is before you want to be able to make extra copies of cells, or this is even before meiosis, when you want to go and create sperms and eggs. So every time you want to go through mitosis or meiosis, the chromosome right here has to be duplicated. Now, let me show you this, what it looks like. So for example, we know that humans, for instance, the human are made of 46 chromosomes. So just to get clear, because you have to understand some of the words. So I'm going to basically line up here 46 chromosomes. That means in that one nucleus, you have 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36, 37, 38, 39, 40, 41, 42, 43, 44, 45, 46. So this is all the chromosomes that are found in the human. Now, this is when the cell is just living its life and it's not going through mitosis or meiosis. However, if you decide that it's time to go through mitosis, you have to make a copy. So when you make a copy, that means you're going to make the sister for each one of these guys. And when you do this, now you can see here that you are adding another 46. But remember, oops, this one here, let me wait. The red one is a copy of the purple one. So basically, each one of these are known as the sister chromatids. So I am making the extra copies because I am going towards mitosis or meiosis. So now I have in total 92 chromatids. Oops, let me find here chromatids. So I have 92 chromatids, but so that means each one of this is a sister. This is a sister, this is a sister chromatid, a sister, and I can keep going, but I'm showing you how this is known as a sister. Now, what keeps them together? That means these are bound by centromere, and I can keep going through writing the centromere here right between each of these sister chromatids. So remember, each one is a sister chromatid. Now this here has now 92, but that's 92 is just before mitosis or meiosis. So moving here, then we come to the word homologous chromosome. What does the homologous chromosome has to do? It has to do with chromosomes that have some things in common, which they are the genes. Now here you can see there's two sister chromatids, right? There is the blue sister chromatids and there's the red sister chromatids, and they are connected by their centromeres. Now, let me explain how these homologous chromosomes has to do with this. So, homologous chromosome, imagine that you have here a gene A, a gene B, and a gene C, and it's connected to its sister chromatid, which is gene A, gene B, and gene C. Now, that's a sister chromatid. Now, you have another sister chromatid at here. But this sister chromatid has D and a D here. It has a C, and then it has a gene here that must be A and a B. Now, these are sisters, and these are sisters. Oops, this is supposed to be A. Right here. And of course, they have to have the same thing. So, sister chromatids. Now, are these two considered homologous? Now, if you really look at close, they are not homologous. Why? Because look, on the first line here of the gene locus, that means the position of genes, this one is a gene A and this is gene D. So they are not the same. If they're not the same, then they're definitely not homologous. However, if I decide to erase all of this right here, right? And instead, I decided to put A here and A here, and I have B here, big B here, and I have little c here and a big C here. Now, if you notice, they have gene A, they have gene B, and they have gene C. That means the sequence of their genes are basically the same. That's when they become homologous. Homologous chromosomes. That's just because they have the genes on the same position. Now, do they have to have the same exact alleles? No. So let me show you here more a clear picture what I mean by homologous. Let's just say this one has big A, big B, and big C connected to his sister, which is big A, big B, big C. And then the homologous can have little a, little B, little C, little C. Are they homologous, these two sisters? Yes, they are. Why? Because they actually have, right here, on the first row, they have a gene A. The second row, they have gene B. And the third row, they have gene C. So it's the same genes at the positions, exactly the same. Now, if you notice, they do not have the same allele, but it doesn't matter because they still have the same type of gene in that sequence. So that means they will be considered as homologous. Now, how would I put homologous in this case scenario? If I would have put this into the case of scenario, it means these guys together will be a homologous, these will be homologous, these be homologous, and keep going. So that's what homologous means, is when you're getting two of the sister chromatids and you're connecting them, because they have the genes location at the same position. So you got, see, this is homologous, homologous, homologous. It's always paired of two sister chromatids and then the finalize. So here, if you want to count how many homologous we have, is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23. So you can see there are 23 homologous, homologous chromosomes here, in which in homologous you have 4 chromatids. So in other words, when we say about homologous are usually made of 4 chromatids, or you can say they're made of 2 sister chromatids. So that means there's 23 pairs of homologous inside a cell. So, but this can only be after you have made the copy of the sisters. If you don't make the copy of the sister, like I was saying, if you don't create the sister as I was mentioned here, the extra sister here, you couldn't make the homologous. So that means you have to have the sisters in order to go from the sisters to homologous. So now that we have an idea like this, let's talk about when it comes to the word diploids and haploids. Diploids, it's a complete set. Okay, it's a complete set of any organism, and usually it's 2n. So for example, a complete set of a human is 46. So if 46 it's a complete set, the haploid will actually be half of the set. And the half of a set, which is n, will be what? 23. That will be the human sperm, in this case, or eggs. Now, a more fancy word, more scientific word, we call it gametes. That's what gametes means. Gametes are eggs and sperms. And these guys can only be produced during meiosis. Now, somatic cells is another word for body cells. And these examples can be the heart, it can be the lung, it can be the muscle. These are all considered somatic cells, and all of them are 46 chromosomes, while the sperms and the eggs, these ones are 23. That's what I mean by haploid. But let's just say if you get, for instance, a species that is, for instance, let's just say 92, that's its diploid. If diploid is 2n, what its haploid would be? Its haploid would be 46. So this is something that are the tricks. Now here are showing all the diploid cells of all the chimpanzees, right? Fruit flies have 8, so that would be considered as the 2n. If I had to find the n of a fruit fly, it would have been 4. The n of a pea plant would have been 7. The n of a mallard would be 40. The toad would have been 11. And you can keep going for all of them as you go through. So that means basically that from the diploid, you take a half of it, that would be the haploid. Now, of course, you got to remember that diploid means the total amount of the chromosomes is a complete set. Now, we usually use the diploids in the mitosis, and the haploids are used in the meiosis. What does that mean? You can always start mitosis, right, with the 46 plus its copy, right? Plus that 46 copy, right? And then you go through mitosis, and when you go through mitosis, in the end, you're going to have 2 identical cells that have 46 and 46, because each one of these 46 were split here. But these are identical to the original one, which is this one. So this is the case of mitosis. When it comes to meiosis, you also have the 46, but you also have the copy. First, they are split into 46 and 46. But over here, they have a crossover happen, and which crossover, it means that there was a mixture of the genes. So these 46 are not identical to the first ones. And then you go through a process of dividing even more, and they become 23, 23, 23, and 23. So these, for instance, now are considered as haploid, while these ones continue to be a diploid. Now, these are usually what happens in meiosis, and this is when you're producing in sperms and eggs. So that's the difference between mitosis and meiosis. Mitosis, you always go from a diploid to a diploid, and meiosis, you go from a diploid to produce a haploid. Now that we're just talking about chromosomes and everything, now we can talk about karyogram and karyotyping. Now, karyogram here is basically using cells, and in the cells we collect all the chromosomes there is inside that nucleus, and then we eventually put them into our alignments, and we see the chromosomes. So karyogram is just about looking at the chromosomes inside the nucleus. Now, how can we pick up those? There's two ways. We can use the chorionic villus sampling, which we basically take a piece out of the chorionic section, or amniocentesis, which we take out of the amniotic fluid. So you can see the needle here. The doctor will put the needle and will try to get some of the liquid here inside of the placenta of the baby. And this liquid is known as amniotic fluid. So when you take that liquid, you take some cells inside of that liquid, then you take the chromosomes inside the nucleus and you organize. So when you take those nucleus out, what it looks here is that it has shown all the chromosomes. So all of these chromosomes are found in the nucleus, and we have them all appeared. Now, this usually happens when we are taking out of the mitosis because the chromosomes can be seen visible. Now, what we have here, we can use these chromosomes that we see here to figure out about their gender and also to figure out if there's any abnormalities. So that's what's the whole point. So karyogram is just to find out what is their gender, if it's a male or female, and to figure out if they have any kind of issues, abnormalities, such as more extra chromosomes than normal, if they have any kind of disease coming up. So as you can see here, all the chromosomes are all spread, right? It's all spread and it's organized. So what we do is we take these chromosomes and we line up them according to their size and structure. As you can see them, they're all matching with the same size. And it came out that all of them were matched really well according to their size and all the length. So their size and structure, this is how you arrange these guys. Now, when you're arranging these guys, this is what usually happens, is that by putting them together, you can see that the 46 chromosomes are paired up. Now, when they paired up, they usually what they do is they end up coming out to having about 22 pairs. As you can see here, they're all 22 pairs that I've made. And they notice that on the last one, right here, the last one, this came out to be the one that is actually telling what's the gender. Now, these ones that you highlight in yellow, these are known as autosomes. And these ones are known as sex chromosomes. So the chromosomes here, we're going to see that if it's X and Y, it's going to be a male. And XX is when it's a female. So here we took out of the fetus baby and we look at the karyotyping. Now karyotyping is the total number of chromosomes and their types in the cell. So karyotyping, as you notice here, is a picture, right? And in this ones, we're talking about the different types of cells. So type of cells here, basically all of these, all the way to 22, all these ones that I'm highlighting, like I said, these were the autosomes, and that's what karyotyping is, the total number of chromosomes and their types of cells. That's what karyotyping is trying to say. And then you have this one, which is basically the sex chromosome. So once again, this is known as the sex chromosome. and this is known as the autosome. And this one you can see there's two X's, so that means XX, which is a female. However, karyogram, this is where students get a mistake. This is just a picture of the chromosomes, and this is just basically used to determine the problems and the gender. So karyogram is basically the picture. Now, telling what type of chromosome, that's karyotyping. So it's very important that you know the difference between karyogram and karyotyping. Karyogram is the picture, and karyotyping is the different types of chromosomes there is in the number. So as I have separated here, the autosomes, right, is usually the pairs 1 all the way to 22. So 122 is the autosome. Now the word heterosomes is the same thing as sex chromosomes, and that is on pair number 23. In this case, you see an X and a Y, and you can notice that the X has a big line, and when the Y is going to be very tiny. So when you see a big line like this with a tiny one, this is known as XY, and this is known as a male. But if you see two big lines, then that's XX, and that is a female. But how do you find this? You always find here in the bottom, the last piece of pair. This is where you find the sex chromosome, and you find the gender. So as it is written here, 22 pairs are autosome, and 1 pair is a sex chromosome. This is when it comes to humans. And again, XY is for male, XX is for female. So that was just to find the gender, but you can also find diseases, for example, like Down syndrome. Now, Down syndrome is a trisomy. When I say trisomy, that means it has 3 chromosomes, not 2. And it's found on the chromosome number 21, so it's the triple X. So you can notice here that there is 2 on number 1, there's 2 on 2, 2 on 3, 2 on 4, 2 on 5, and you can keep going, and you can see that it's always 2. As long as it's only 2, that is normal. However, you reach on chromosome 21, and you don't see 2 anymore, you see 3. And that means this is where the problem is, because as humans, we're only supposed to have 2. But if we have 3 or just 1, then there's an issue. In this case, we saw that 21 has 3, so that means every time you see 3 on number 21, on chromosome 21, that means you have Down syndrome. And you can see 22 is normal, right? Again, I'm putting blue, it's normal. And here, if you have X and a Y, it's a male. If it's XX, it's a female. So it depends on the situation. You can find the gender. But, like I said, the chromosome 21, it's Down syndrome. Now, if by any chance you actually had 3 on 14, that doesn't mean it's Down syndrome. Down syndrome can only happen at 21. So it's very important that you understand that many students think, oh, if I have 3 here or 3 here, that means it's Down syndrome. No, Down syndrome can only happen at 21, on the pair of 21. So what is this Down syndrome again? It's 3 copies on chromosome 21. This is caused by non-disjunction. What does non-disjunction mean? If you remember in mitosis as well as meiosis in this case, so mitosis basically we have prophase, and then we have metaphase, and then we have anaphase, and then we have what we say telophase. Oops, let me write this again. Telophase. If we remember prophase, they're becoming visible, and you have the spindles coming, and then metaphase, you have them lined up in the middle, and then anaphase, that's where you start to separate them, and you can see that I'm equally separating them, and then telophase, you have the nucleus coming back, and the chromosomes become uncondensed. This is a very quick one, because you can learn this in the topic 1.6. So, non-disjunction means, as you noticed here, on the anaphase, you divide them equally. That means 3 went to one side and 3 went to the other side. However, non-disjunction, you might happen to make a mistake. The mistake will be that you end up, in this case, maybe sending 4 to one side and 2 to the other side. So you can see right here that it's uneven, right? And the reason why you see it's uneven, because as you notice, you have 4 going to one side and you only have 2. When this usually happens, by the end of the cell, this one will have 4 and this will have 2, and that means it's unbalanced. In the same case as if you are trying to create cells, so for example, if you are making sperms and eggs in meiosis, for instance, which also has anaphase and telophase, once we get to topic 3.3, you get to learn about meiosis. is that when you're creating sperms, sperms are supposed to be 23, right? And the eggs are also supposed to be 23. So you have 23 chromosomes on the sperm and 23 on the egg. And when they join together, they make the 46, which is a human. However, because there was non-disjunction happening during meiosis, where specifically at the anaphase, they did not separate the chromosomes equally. So that means one chromosome ended up having to be 24 and the other one might have to be 22, or it could have been 23. In this case, if these sperm and egg join, what happens? They are 47. So you can see the human needs to be 46, but there's this extra one chromosome. That extra one chromosome, if it happens to be on chromosome 21, then that's it. The child has Down syndrome. And this is because, again, one egg from mom came with 23, or it can be the sperm that came in with 23 and the other one came up with 24. So either case, doesn't matter if it's the sperm or the egg. If one came with extra one and you get 47, if the extra one is on chromosome 21, then automatically it's Down syndrome. So here is one thing that IB will do is give you some karograms and they tell you figure out what's the gender. So you look in the bottom here and you see there's a big X, big line here, chromosome, and it's tiny one. Obviously there'll be X and Y, which that means this is a male. So whenever they ask for the gender, look at the last pair. Then here is also another example, and they can say, oh, if they say, look at this karyogram and figure out if there is any diseases and what's the gender. So the gender, again, you go to the bottom one and you notice that this has two exactly the same size, and that is XX, so that's a female. And then you look in each one of the chromosomes, you see there's 2, 2, 2, 2, 2, and you keep going. You notice that all the 2s are happening for each chromosome number, and that means that it's normal. However, you reach here and then there's 3, and you notice it's on 21. Automatically, you know that this is Down syndrome because you only supposed to have 2, but now you have 3. So this is a female that has Down syndrome. So now you understand about karyotyping as well as karyogram, and that karyotyping can happen a lot coming in the exam. So it's important that you know how to read this. Remember, if there's more than 2, that means there's a disease. If it's on a 21, it's Down syndrome. And whenever you want to find about the sex chromosome, look at the last one and see if they have the same size, it's XS. If one is bigger than the other one, like in this case here, then it's XY, then that's a male. So now we're going to talk about Cairns technique, and this is to measure the length of DNA molecules using the autoradiograph. Now, the interesting thing about Cairns is that he was able to figure out the true size of the DNA. See, because we couldn't figure out the size of DNA when DNA is coiled, right? Because when it's coiled like this, right, then it's visible to us, but that's the only time that we can see it like a true chromosome size. But is this truly telling us the size? No, this will just tell you what's the size here between this to this, the length of this one. But that's not what Cairns did. What he truly did was able to open this entire DNA like a strand completely open, which is so huge, and he was able to find the real size of the DNA length. So here's a picture just to show you. So you can see that this chromosome here has been coiled, right? And what's inside? You actually have those proteins that it's inside that it's coiled around it, right? Because DNA is coiled around the histones, as I mentioned at the beginning of this video, right? It's like you have a protein here, and then you have the DNA wrapped around like this. Only that way that you can see a visible chromosome, because again, it is coiled around the protein, and now it is really visible for us. But when it's coiled like this, it's not really stretched. So if you look at this, can you actually know the size of this line? No, you can't, unless you take this and you stretch it completely. If you stretch it completely, you might take more than this computer line, right? So that's what Cairns did. He looked through the entire size and not the size of when it's coiled. So he looked at when it's uncoiled, what exactly is its true size. Now, how did he do this? He basically, since we know that DNA is made of A, T, C, G, G, and keep going, you can put whatever letters you want here. What he did was he highlighted the T's. And when he highlighted the T, he had a better chance of figuring out the true size of the DNA. because let's say the DNA is this huge, whatever is the last T here is, or whatever the last T is here, now you can get the true size of how big the DNA length is. So here is the experiment, and you have to know how the experiment happens. First, what he did was put a bunch of radioactive thymine, which is the T that I'm talking about, that will be the T that represents of the A, T, C, and G, inside a cell. Now, that T eventually got into the chromosome of the DNA. So you can see that you have the radioactive put it inside cells, and then the cells were broken down. Once you've broken down the cells, you took the chromosomes and you isolated them here. So that means you took all the chromosomes of the nucleus and put them into this one slide right here. Then after you did that, you basically put inside a very important emulsion, which is a silver bromide. So that means you take these slides and you insert it into this liquid. Now this liquid, again, is made of silver bromide, and you have to remember that. So once again, just beginning of the process. First, you put the radioactive T inside here. The cells took those T and put them into the chromosome. Then you open the cell and the nucleus, and you got the chromosomes inside the nucleus. Then you put all those nucleus in a slide. You separate them all. Remember that these chromosomes have T now, highlighted T. Then you put them inside this bucket, and inside the bucket there is a liquid that is made of silver bromide. Now, here's the most important thing. Every time the silver, right, of the silver bromide reacts with the thymine, it becomes into metal. So that means when the T, the highlight I'm going to put here as highlighted T, when it reacts with the silver, Ag is silver here, it turns into metal. And the other parts that don't have T, it just continue to be a liquid of silver bromide. So here you can see that the parts that have T, as you can notice, these are the highlighted T's, right? These highlighted T, they reacted with the silver. And the ones that did react with silver, look, they became black, which means they became, I'm putting the green here, they became metals. What about the rest? The rest did not react with T. They continue to just being a liquid made of silver bromide. So if the Ag touched the highlighted T, it becomes metal. The rest of the AgBr continue to just be liquid, okay? Now, what do you do with the rest of the liquid? You just washed it away. So in the end, what do you have? You only have the metal part, the black dots, right here, as you can see. Now that you only have those black dots, as it's shown here, you can see that they're just black dots. You put them in a photography film, and they're developed. And then what happens is, when you have an X-ray hit on the black dots, right? Remember the X-ray, if you have an X-ray hit on things that are dots like this, anything that is metal, what happens? If that light hits on them, that means you're going to see a shadow of them right here, right? The exact shadow, these are the shadow of the metals that were placed here. And of course, the X-ray was hitting on these metals, and it projects like that. So this is what we see. We saw that, and then from there, we developed a film, and eventually, this is what we have, autoradiography. And now we have the true size of the DNA length. And all because of what? Because all of these guys here, all of these metals here are actually what? All of them are the T's that were highlighted that mix it with the Ag, with the silver. So depending on the DNA structure, you can find a T here, and the edge might have some A's and G's here, but at least you can have a better sense of what is the actual length of DNA, which you didn't have it before. So the great thing about this Jordan scan technique is that it was able to show you other things like the chromosome replication. It was able to show the replication bubble, and as well as bidirectional. That means you can see that you can open the helicase on both sides. From this picture, you were able to see the helicase can go in this direction as well as this section, which means bidirectional. So you can also see here the pictures, how they were able to figure out the DNA replication and how the bubbles will open and create new DNA. And last but not least, when we talk about database, which is the last section of 3.2, is talk about the gen bank. Now, what it do? It just basically look at collection of DNA sequence, and you can also find the polypeptide products of a certain gene. So if you inject that gene in there, like a gene number you put in there, they will tell you what are the types of proteins that can come out of that gene. So the gene bank usually has a huge list of all the species with their chromosomes, sequences, and basically their DNA-based sequences. And then you can just throw a sequence in there and say, you can figure out, hey, where does that sequence come? Oh, it comes from this species. The best part also is that you can throw in there a gene, and from that gene, you can figure out also what are the kind of proteins that can come out of that gene. This is all done with the gene database, the gene bank. And this is pretty much for 3.2. Here are all the study guides. These questions can be answered through this video. If you need to go back to the video. You'll be able to answer all of these questions as study guides. These are also, if you go through these study guides, it really gets you strong and ready for the test. Thank you.